It is my great pleasure today to introduce Angela Dorigatti, and she is a spatial field application scientist at Takara and formerly Curio Science. And just as a side note, prior to that, she was also at 10X, so she has a lot of experience in this molecular space. Today, Angela will talk to us about tracker single-cell spatial mapping, as well as seeker spatial RNA-seq mapping. And with this, I give it over to you.
Yeah, thank you so much.
So welcome, Angela.
All right, thank you. All right, so my name again is Angie Dorigatti. I am from San Francisco, so I'm really happy to be here today to tell you guys about the newest of technology called Tracker. And so just a little background about Takara Biosciences. They established as Culinotech Laboratories back in 1984. In 2005, they were acquired by Takara, and they have been since working in the single cell game ever since. So in 2017, they acquired Rubicon Genomics and Wafergen Biosystems, which allowed us to understand single cell biology at a molecular level. And just most recently, we've added Curio Biosciences to our portfolio with the newest spatial technology. So today we're going to be discussing the Seeker and Tracker kits, which are available through Takara and also through Scientifics, who is the distributor here. There's a lot of single cell has definitely evolved over the years, and we have a lot of different types of single cell ways to look at single cell information. Two things that I want to point out is the CytoSeq, which the original publication is what became now known as BD Rhapsody. And this was previous work that was done by some of the Curio co-founders, and also DropSeq as well, which also gained a lot of traction in academia, which was published by Evan McCosco, who was one of the founding pioneers of Curio as well. And so the two groups of single cell pioneers in both CytoSeq and DropSeq joined forces to form Curio Biosciences. Now, we have a lot of different ways that we look at single cell. And so, you can use several different techniques. You have the cell per well. So this is like the isolate or the Shasta system. There's the droplet base. So you're thinking about 10X genomics or combinatorial indexing, which is parse, or there's a couple of other ones. But in the end, what we end up with is uniquely barcoded single cells or single nuclei. And from here, there's been so many different technologies that have now been enabled for this. So we have the transcriptome, chromatin accessibility, histone modifications, and also DNA methylation. But one issue that we have is when we begin to analyze this data, what we see is this beautiful UMAP, which gives us all the different types of cells. But then we need to know, where did these cells come from? Structure and function are directly related. And so without this structural information, it can sometimes be very difficult to parse out what's happening with this data. So currently there are some technologies that are available for spatial. One is a sequencing-based assay. This would be like our Seeker technology or the Visium assay. And so what you do is you cut your tissue, you put it down onto a slide, and then you release the mRNA onto the slide. And then we sequence the mRNA that's on the slide to figure out where those transcripts came from. The problem with this is when you cut a tissue, place it on a slide, you're not releasing 100% of that mRNA. You're only capturing about 10 to 15%. And so when we're used to single cell, we're used to seeing hundreds to thousands of transcripts per cell. When you move into the Visium world, you start losing that sensitivity. So you end up getting dozens to hundreds of genes per cell. You also have the problem that when we cut the tissue and release the mRNA onto the slide, we have these discrete spots, but we don't know exactly what cell it came from. We just kind of have to estimate, is this what cell this came from? So if you have a neuron and an astrocyte that are overlapping each other, and both of them release their mRNA down onto that slide, you're going to have a spot that has both transcripts for neurons and astrocytes. And it's actually very difficult to deconvolute that information to figure out what transcripts came from which cell type. In the end, you'll usually end up calling one of those spots either a neuron or an astrocyte, but there's going to be contamination of other different transcripts from different cell types that's going to muddy the data. We also have microscopy-based assays as well. So think of like the xenium or even doing something as simple as FISH. But you're also limited by the number of probe panels that you can use. You can also be limited by the number of fluorophores or how good your antibody is. But it is another tool that you have in your toolkit to understanding the biology of your samples. And so if your resolution is not high enough, you have to do some segmentation or binning. If the resolution is too low, then you end up having to do some deconvolution. And so what we really want is the single cell level. We want to know what's happening in each one of those cells. And so Evan Makosko and Fei Chen over at the Broad Institute came with their pivotal paper that discussed the technology of slide tags. And slide tags allows us to give a spatial barcode to each cell prior to sequencing using the single cell technology. So now we have the first true single cell resolution for your kit. It plugs into a standard single cell workflow. It converts your spatial data or single cell data into spatial, and it has the same sensitivity as your single cell workflow of choice. So we can now take your mu map with all the clusters and bring it back to a two-dimensional spatial location. So how does Trekker work? We have a tile, and on this tile, there is a glass slide. And on the glass slide, we have a monolayer of beads that each bead has its own special, unique barcode for the location. And what we'll do is we'll cut the tissue, put it on top of the slide, and instead of releasing the mRNA down onto the slide, we're actually going to release barcoded oligos up into the tissue, and then we're going to perform a nuclei isolation. Now, because we're working with fresh frozen tissue, we can't get whole cells, but we can isolate nuclei, which represents usually about 40% of the total RNA that's happening within the cell, and it correlates really well with what is happening within the cell. Part of this technology also includes a photocleavable linker. So this is how we release the oligos from the beads and make them diffuse up into the tissue. So we put the tissue on the tile, we put a UV lamp on top of the tissue, and the oligos then are cleaved from the beads, and then they're released into the tissue. From there, we do a nuclei isolation, and then you'll end up with two different libraries. You'll end up with your regular gene expression library, and then you'll also end up with your Trekker spatial library. Now, the nice thing about this assay is that it is fairly quick, very easy to do. It only takes about an hour upstream of getting your single nuclei suspension. And once you get that single nuclei suspension, you can treat this like you would any other single nuclei. So you can use it as input into your 10x genomics or your BD Rhapsody. We're also enabling parse, and there's a few other ones that we are also enabling as well. So here, so the great thing about this is that there's no special instrumentation that's needed. It's a simple reagent case kit solution, and you can use it upstream of the workflows that you're already working with. So here's a little short video on how this Trekker slide technology works. So we have our Trekker tile, which includes the barcoded beads that have a spatial location. We melt the tissue on top of these beads, use a UV light to release the oligos up into the tissue. And then we remove the Trekker tissue, or we remove the tissue from the Trekker tile, and then we dissociate them into single nuclei. From there, you can take those nuclei and use as an input into several different types of assays. So you could use a well-based plate assay. You can also use droplet-based or combinatorial indexing. In this case, we're going to look at the droplet-based assay. So the Trekker barcoded oligos will be taken up by the bead that is available from 10x Genomics, along with the mRNA. and you'll get two separate libraries. You'll get the gene expression library, and then you'll get the spatial context along with it. Now, we don't expect that just one bead is going to donate all of its barcodes to one nucleus. We do expect to see that there's going to be some diffusion. And in this case, diffusion is actually working for our advantage. So the bead that's directly below the cell is going to donate the most amount of its oligos. The beads that are farther away are going to donate fewer of the oligos. And we use that ratio to figure out exactly where the original location of that nucleus was within 2 microns. This is a little bit different than the regular slide-based captures, where diffusion is actually working against you. In those cases, you're releasing the mRNA down onto the slide, and any lateral diffusion can just add a lot of noise to your data. So we release the barcodes up into the tissue. The spatial barcodes adhere to the nucleus, and then we figure out what ratio those barcodes are in order to figure out where that original location was. It's kind of like doing a GPS triangulation using this Gaussian distribution. And with that, we can figure out where the position was within 2 microns. The great thing about this, though, is that now you can superload because we can detect doublets very easily. So because we're barcoding each one of these nuclei before we put them through the system, we can now confidently detect, is this 2 small cells that came from 2 different locations, or is this 1 large cell that's from the same location? And now, while we can't rescue that data, we can confidently remove it and clean up the data even more. This is a destructive process, so we're working with fresh frozen tissue. There isn't enough time to do an H&E staining and take some imaging. So we do recommend that you take an adjacent section and do an H&E staining on it. And on our website, we do have a program that will then align those 2 images on top of each other. So then you'll have the morphological data along with the gene expression data with that spatial context. We do have a 10 by 10 tile, so it's about the size of a mouse brain. It is true single cell resolution, so we're no longer limited to how many spots we have on our tile. The tracker barcodes are just released up into the tile or into the tissue. If we had smaller or larger, it would just help with the triangulation. Everything is still going to be a single cell resolution, though. The nucleus is still its own discrete package of information. So even if we have 2 cells that are on top of each other and they end up getting the same spatial barcode, it doesn't matter because we're removing those and we're looking at them individually for their own information, own gene expression information. Currently, we are only compatible with fresh frozen tissues, but we are enabling FFPE as well for down the road. And there is no specialized equipment. All you really will need is that UV lamp, which is just a one-time purchase. You will need a cryostat, though, and someone who is an expert at cryosectioning. One of the hardest parts about the assay is just cutting the tissue and fitting it on that small tile. You'll also need a downstream platform, so you'll have the tissue cut, then you'll use the Trekker kit to isolate the nuclei and to barcode them. And then you'll need your single cell platform, so your 10x Genomics or your BD Rhapsody. And then you'll also need to do sequencing as well on top of that. The throughput is about 2 to 4 samples per run, but it only, after you put the tissue down onto the tile, it only takes about an hour for the entire protocol. And this is a really important matter because a lot of times nuclei isolations can take 2 to 4 hours to do. Because we're working with such a thin slice of tissue, the dissociation time is a lot quicker. So you end up having not as much cell type bias with these that some of the longer dissociation protocols can have. Sometimes there are cells that are more fragile than others that tend to lyse a little bit easier, but because the entire dissociation time is actually only between 10 and 20 minutes of the cells sitting in the lysis buffer, we end up preserving a lot of those rare cell types. And it's the same sensitivity as their single cell workflow. So if you've done single nuclei RNA sequencing on your cells before, the data is not going to look any different just by adding those Trekker Legos. We have now enabled a whole bunch of different types of assays. So we have the BD Rhapsody, the Illumina single cell 3' RNA Parse Evercode, Scale Biosciences, and 10x Genomics 3'. We also have recently released our 5', and we also have TCR, BCR, and also attack sequencing and 10x multiome that's enabled. So once a lot of these technologies moved to those slide-based assays, we lost a lot of these multiomics. Now we can use all of these multiomics again. So one example of this is we took a 25 micron mouse brain section, and we were able to identify all of the major cell types. There is no deconvolution or cell segmentation that needs to occur. And we were able to map that back to spatial locations, and we were able to see all of the expected cell types. With we ran about 30,000 nuclei. Each one of these little dots represents one nucleus, and each one has a gene expression of several thousand genes. So how we've used this is now we can look at the finer structures of the hippocampus, the cortical layers, the thalamus, hypothalamus, striatum. And we can also look tease more into microglia. And so one thing we did was to take a look at the different cortical layers. Just looking at this UMAP, we know that there's a bunch of different cells that correlate to different cortical layers. But once we map it back, we can see that each one of these cortical layers has its own gene expression that is dependent on where that cortical layer is in two-dimensional space. Another way of looking at this is we've got this UMAP, and we have two different astrocyte populations. We have this green astrocyte population and a pink astrocyte population. And just looking at the UMAP, we know that there's differences in these two different populations because they're clustering differently. But just looking at the UMAP, it's very difficult to figure out exactly what's going on. Once we map that back into space, now we see that the green astrocyte 1 population is more towards the center of the tissue, whereas the astrocyte 2 population is kind of sprinkled all throughout the brain. So what we wanted to understand is what is happening with this astrocyte 2 population and what makes this astrocyte 2 population different. And so what we decided to do is take a look at the astrocyte 2 cortical layers. So we took all 6 of those cortical layers, and we were able to see that the gene expression in these astrocytes changed depending on what layer they are located on in the cortex. Without having this spatial information, figuring this kind of information out would be very, very difficult. And by adding the trucker oligos, we don't actually impact anything. And because we're using single nuclei as opposed to relying on sequencing a slide, we now can get way more gene coverage than you're used to. So instead of dozens to hundreds of genes per cell, we're now seeing hundreds to thousands of genes per cell. If you've used 10x Genomics before, you might notice that when you get your UMAP back, instead of having a lot of discrete puncta for the different clusters, it ends up looking more like a continuum. And that's because if you have multiple cell types that are donating mRNA to a single spot, you have to call that spot one cell type versus the other. And so the noise is the data is kind of noisy. Because we're working with single nuclei, we see those distinct, discrete clusters now. And so it's much easier to figure out what's happening in each one of these cell types because we have all of the information. So now your question becomes, how many nuclei then do I load into my 10x or my BD Rhapsody? Well, normally when you're doing single cell, you'll take a chunk of tissue and you'll dissociate it. And so in this case, we dissociated a 3 milligram chunk of brain tissue, mouse brain tissue, and we got about 3 million cells. 10x Genomics recommends that you load 10,000 cells. And so that's what we did. If you were to look at a single plane, that map is actually pretty sparse. You really can't make out much. But because we're working with a very thin slice of tissue, now that 10,000 cells is much more impactful. So here we took a 25 micron slice of the tissue, and we were able to extract 150,000 nuclei. And from here we ran 10,000 cells, which turned out to be about 7% of the total nuclei that were isolated. And now you can start seeing the finer structures of the hippocampus. You can start seeing some of the cortical layers coming out. And then if you decide to run even more nuclei, now the density of your data is even better. Now you can start seeing even the finer structures, and you can start doing some of those cell-to-cell interactions. So in this case, we loaded 30,000 cells, which turns out to be about 20% of the total nuclei. So you don't need to run 100% of your nuclei. You can run a subset and still get the same type of information. You could, though, if you wanted to, you could run 150,000 cells if you got 150,000 cells. You would just have to run them in separate lanes and then combine the information at the end. But that does get quite expensive. So for 10x genomics, because we can superload for the new GEM-X chip, you can load up to 50,000 nuclei per well now. For the BD Rhapsody, you can load up to 100,000 nuclei. So you could load 3 different lanes with 50,000 nuclei and sequence them all. But again, the sequencing costs for that would probably be pretty expensive. So you could just do a subset. And most people decide to run between 20 and 30% of the total nuclei that they get, or between 20,000 and 50,000 total nuclei. If you're running something small, like a punch biopsy, and you only get 10,000 cells, then you would just want to run them all. The nice thing about the Trekker tile, though, is that you can put multiple samples onto one tile. So if you have smaller pieces, you can put several different samples onto one tile to maximize your costs. And then what you can do is when you get to the data analysis, you just put a loop around each one of those tissues, and you can label them, and then you can analyze them that way. So now we're no longer limited to just having a gene expression only with our spatial information. We can now use it for coming up with something like ATAC or VDJ libraries, and we can put it all together into one sample. So for example, we took a metastatic melanoma tissue, and we ran both the single nuclei RNA sequencing, we ran ATAC-seq, and we ran TCR enrichment, and we were able to get robust information from all 3 of these assays. One thing I do want to note, though, is that because we're working with nuclei only, the most finished clonotypes for TCR, BCR are in the cytosol, and we're only running nuclei and not any cytosolic transcripts. So it's not going to be as robust as when you're running whole cells, but doing this project, we were able to find that you are able to at least find some of these different clonotype information. And when we add the Trekker oligos for barcoding into the tissue, it actually doesn't change our data. So when we look at the ATAC data, it overlaps exactly with what we were with Trekker tags or without Trekker tags. Once you get your data back, you have a couple of options for bioinformatics as well. So on our website, we do have some a workflow chart that will kind of go through each one of the steps that you can use in order to remove the background, doing some filtration, doing cell type and disease state annotations. But we also have some other options for you. So your first option is to run the pipeline locally. So you can install the pipeline locally and run it yourself. What you'll do is when you end up with your final libraries, you'll have 2 different libraries. You'll have your gene expression library and your spatial library. You'll take your gene expression library and you'll put in whatever pipeline that was created with. So like 10x genomics, you'll run it through you'll take the FASTQ files, put it through the 10x genomics pipeline. And then you'll take your Trekker library and you'll put the Trekker library through the Trekker pipeline, and then you'll also add the outputs from the 10x genomics pipeline into the Trekker library. We've also partnered with a couple of other groups as well for data analysis. One is called Latch Bio, and the other one is Biotouring. Biotouring is kind of making waves now. They have some really easy ways of doing spatial analysis. They have their Spatial X platform, where you can do a bunch of different types of secondary analyses, not just your primary analyses. And we do have a workbook that's available through them to run the entire Curio pipeline. Latch Bio is also another option that you have. This one is you pay for how much you use rather than a single license, which BioTuring requires. And this one is currently being used by biopharma and lots of different R&D labs throughout the world. But it's very easy to use. So if you're not very good with bioinformatics, or if your bioinformatics is taking a very long time, you could do the analysis yourself. You just need to upload your data, and there's several different ways that you can upload it. And then from there, you can go ahead and select the pipeline that you'd like to use. And again, we have our pipelines already preloaded into their workbooks. And then you can, it's a simple, easy interface where you can basically point and click to do different analyses, such like Excel by gene, which will allow you to toggle between your UMAP and your spatial gene expression. So now we have a kit that achieves true single cell resolution while preserving the high sensitivity and gene coverage that you come to expect from single cell. And now we have also re-enabled the multiomics, which was once lost. And this fills in the missing spatial context without actually changing the experimental and computational infrastructure. So whatever you're using for your single cell analysis, you can continue using that in order to analyze these kinds of data sets. You don't have to do any complex cell segmentation or deconvolution to help understand your biology of your sample. Everything is still in that discrete single cell package. And so it makes data analysis take weeks instead of months. And that's good science. All right, so Nikki is here as well. She's from Scientifics, and so she's here to help you with any of your sales questions. If you guys have any sample questions or any technical questions, I'm more than happy to help you. So if you guys have any questions, I can open it up to you now. Yes.
Just in terms of how it works, so you know in advance where all the beads are on the slides.
Yes, each slide is unique. So each slide has a tile ID. And actually what we do in manufacturing is that once we've created each one of the tiles, we put it through flow sequencing so that we can know where each one of those, what each one of those beads has as their barcode, so that we can use it to map back. So you'll need to make sure that you keep the tile ID, and then we have a barcoded whitelist that you can pull from. Yes.
From how many % of the cells do you think you can isolate the nuclei? Like how much is it get lost? You don't obviously get one nuclei per cell, so some of them.
It depends on what tissue type you're working with, but with most nuclei isolations, we do expect to have about a 50% loss. So what you can do is you can do a DAPI staining or H&E staining, and you can count how many nuclei you have in your whole tissue. Then you can do a nuclei isolation on that tissue, and then count your nuclei and see what kind of loss you would expect to see for your particular tissue type. So if it turns out that you have a very less dense tissue type that you're working with, such like adipose tissue, we do recommend a minimum of 25 microns, but you could go up to 30 microns if you want to capture more nuclei. The reason we have the minimum of 25 microns is that most nuclei are 10 microns. And so if we make it the slice only 10 microns, you end up shearing a lot of the nuclei on either side. And we want whole intact nuclei as input. So we found that 25 microns seems to be kind of that sweet spot. But you could go up to 30 in order to get more nuclei. Yes.
How much sequencing of the spatial library do you need to do?
It's very low. So 10x Genomics recommends that you sequence the gene expression library between 20,000 and 50,000 reads per nucleus. For the checker library, you only need 5,000 reads per nucleus. If you're using the BD Rhapsody, it's a little bit more sensitive, and you only need 1,000 reads per nucleus.
So you showed some brain stuff. Presumably the bigger cells are underrepresented because of what you just said about the size of the sections. Is that fair?
Depends on the nucleus. So most nuclei in the brain are under 20 microns. And there's a cell straining step that you can use to help clean up your sample. And so if you're expecting to have larger nuclei, like some adipocytes have fairly large nuclei, we don't want to size select against those. So you would use the larger cell strainer, like a 40 micron cell strainer, instead of a 20 micron cell strainer that we normally recommend in our protocol. But you know, if you have something that is a nucleus that's very large, it could be possible that there is a size selection because of that thickness issue. So you just want to make sure that the most of your nuclei that you're expecting to see is going to fall within that. If it turns out that you have larger nuclei, you could always go a little bit thicker, and that could help with that. Now, if you've ever done a nuclei isolation, you know that it varies tissue to tissue. Not all nuclei isolations are the same. So we do have a couple of protocols available on our website for some different types of tissues. So one is brain. Brain is fairly easy to dissociate, but it also has myelin, which is a very sticky protein, and it ends up trapping a lot of ambient RNA, which can lead to a lower fraction of reads of cells and a lot of noise in the data. So we have a protocol available on our website that uses the Invent Minute Kit to remove the myelin from the sample before you load them. The other one is muscle, heart, tumor tissue. Sometimes those are more difficult to dissociate. So we also have a protocol for tough tissues, and that includes a pestle grinding step. So you'll, once you blow the tissue off of the tile, you'll take that tissue and put it into an Eppendorf tube, and then you can use a pestle grinding step for 3 minutes. And then we also have a recommendation if you're working with something that's heavy in collagen, like heart or muscle tissue, to spike in liberase, which has 2 different collagenases and a protease, but that'll help hasten the digestion process, and it'll liberate those nuclei a lot quicker.
So how close are you to having a version that works with fixed tissue?
We're hoping to have it released before the end of the year. And the nice thing about the FFPE protocol, it's going to be called Trekker FX for fixed. In the end, we're going to be using the 10x Genomics Flex Kit as the final assay that you'll be putting the nuclei through. But for that particular assay, the great thing is that you don't need to do a serial section because it's fixed tissue. We do have time to image and do an H&E staining. So you can do the same glass slide. And so you can section and place on a plain glass slide rather than on the tile. You can do the H&E staining or IF staining on that particular tissue. And then we use a sandwiching instrument. It's way less complicated than the CytAssist. It's way easier to use, and that will sandwich the oligoslide to your tissue, and then at the end you can store your nuclei. So with fresh frozen, we don't recommend that you store your nuclei. At that point, it's very fragile. The Trekker fresh frozen protocol is very quick, though. It's only an hour upstream of loading. So, you can do that workflow easily in a day. But if you want to multiplex, if you want to add several, save up a bunch of samples and run them all at the same time, then the Flex protocol is going to be your best bet. And we're going to have 2 different versions of the Flex protocol. The first one is you're working with FFPE tissues. And then once you get to the nuclei stage, then you load them according to the Flex protocol. The other one is that you just run the fresh frozen protocol, and at the very end, then you fix the nuclei, and then you can store them and use them for later. Yes.
Do you think people will stop using Visium?
That would be nice. I think it's an older technology, and I think that there's just some bioinformatic data. People mostly get stuck on the data analysis. It's not that it's a bad technology. It's just that it's not optimal for understanding what's happening at the single cell level, because there's so much ambiguity with what to call a spot and how to bin it. You also implicitly adding some bias to it. So here, just going back to the single cell idea, I think is going to be the future of what people are going to be looking for.
Yeah, except that you can look at every part of the section, so you're not missing anything.
That's true.
Trade-off, like you can choose how much to sequence across the whole section rather than choosing how many things to sequence.
Yeah. So with Trekker, either if you're doing the FFPE version or the fresh frozen, we are only working with nuclei. So if you're interested in cytosolic transcripts, Seeker is going to be a little bit better. Seeker, we do see about 3 times more genes than we do with the Visium HD assay, so it is a little bit more sensitive, the Seeker is, and that one you will collect all of your transcripts, but you're not going to get the same depth. So there's always going to be a trade-off. Do you want to have a little bit of everything, or do you want to get more in depth into most of the things?
The question.
Yes.
So SLIDE-seq has been shown to be compatible with DNA sequencing. Are you guys working towards that?
Yes, we are. So we don't have anything to release yet, but that is something that is definitely on our radar. Great questions, guys. Anything else?
How thick can you go in terms of tissue sections? Because you say like you can go like more than like up to 30. Can you go even higher if you want to get a little bit more? Like what's the limitation there? Like infusion of oligos, number of oligos?
Yeah, we're not sure about the accuracy of the oligos when you're going higher than 30 microns. We've only tested 30 microns, but talking to our R&D, they say it's certainly possible to go thicker if you need to. You can also do, if you have like a monolayer of cells or a cell culture or organoids that you want to do, sometimes it can be very hard to freeze and embed a single layer of cells. So we have had customers just take their cell layer, plop it on top of the tile, and then they just need to freeze it. And that freezing will help ensure that those Trekker oligos are cleaved properly and get into the tissue. And so we've had great success with customers actually trying fresh tissue, putting it onto the tile, and then flash freezing it, and then moving forward with the protocol. All right, guys. Well, thank you so much. We do have some snacks and stuff that's next door. And Nikki is also here. She's more than happy to discuss with you some more information about it. So if anybody has any additional questions, I'd recommend we go on ahead next door and we can get a little bit deeper into the technology if you'd like. Thank you very much.